Hello everyone. The next topic which we will be studying is about the enzyme action. So before studying the enzyme action, we first understand what are enzymes. So enzymes, they are proteinaceous molecules that help in the catalyzing the biochemical reactions in a body. So due to this property, the enzymes, they are called as the biocatalyst because they acts in the biochemical reactions as well as it plays a catalyst that accelerates specific chemical reactions in the biological system. So that is why these enzymes, they are known as the biocatalyst. Now it alters the rate of reaction in the biological processes. They have high molecular weight that is made up of mainly of chains of amino acids that are linked together by the peptide bonds. So as they are proteinaceous in nature, they also possess secondary and tertiary structures. So when the enzyme, they are present in their tertiary structure, their protein chains get folded upon themselves, and due to this many services are formed, and this is known as active. So these active sites are mainly responsible for the mechanism of enzyme action and also the mechanism of enzyme catalysis. Now we will talk about is the enzyme action here. So enzyme action, they have been hypothesized uh, into two categories. Now there are two categories, that are the lock and key hypothesis and the induced key hypothesis. So these are the two categories of the enzyme action. Now first we will be discussing about is the lock and key hypothesis. So this is a diagrammatic representation which is showing the key, which is a substrate, and there is a lock which is represented as enzyme. So here what is happening is the enzyme, they have an active site and the substrate binds to it. So this is called the enzyme substrate complex. So when there is an enzyme which is enzyme catalyzed reaction which occurs in the two steps. So these are the two steps. In the first step, what is happening, the enzyme molecule and the substrate molecule, they collide and react to form an intermediate compound which is called as the enzyme substrate complex. So this is the first step in which the enzyme substrate complex has been formed. So once the enzyme and substrate complex is formed, the enzyme is able to catalyze the formation of the product when it is released from the enzyme surface. So the hydrogen bonding and the electrostatic interaction which holds the enzyme and substrate together in the complex. The structural features of functional groups on the enzyme, they basically participate in these interactions that are located in the cleft or in the enzyme surface. So this pocket where the enzyme combines with the substrate and it transforms the substrate to a product, this is known as the active site of the enzyme. So this is the early model which is describing the formation of enzyme substrate complex, which is called as the lock and key model. So this model was being proposed by Emil Fischer in the year 1894. So this hypothesis was only to understand the mechanism of action of the enzymes. So the enzyme and the substrate, the molecules, they exhibit various geometrical shapes, and these shapes are very specific, just as the lock and key. And this hypothesis states that the active sites of enzyme act as a lock, that are specific molecules such as COOH or SH, and these enzyme molecules can only be opened with the help of specific substrate complexes. So this model is only to make us understand that how the mechanism or action of an enzyme is being taken place. So there is a enzyme substrate complex at the end which is being formed in the second step of the lock and key hypothesis. So by this hypothesis, it can be concluded that a very small amount of enzyme can act upon a very large amount of substrate molecule. So it can act upon large amount of substrate molecules. And it also explains how the enzymes are not used in the reaction and can be used again and again. Moreover, it helps to understand the mechanism of competitive inhibition as well. So the next model which we will be discussing is about the induced fit hypothesis. Now, Koshland, he proposed this hypothesis in the year 1916. So this hypothesis is actually quite different from the previous hypothesis, that was the lock and key hypothesis. This hypothesis states the active site of the enzyme is flexible in shape and can change its shape according to the nature of the substrate. So it can change the shape according to the nature of the substrate, which means that it can form its active site complementary to that of the substrate. So it can form its active site that is complementary to the substrate. So this model is very easy to understand it in a way that how a hand induces a change in the glove. That is the same way an active site when induces a change in the chemical substrate. The substrate gets into the active site of an enzyme, and according to this, the structure of the active site of an enzyme is flexible. So here you can see there is a domain 1 and there is a second thing, that is, the first one is the open domain. The domain 1 and domain 2 are open, and in the second one it is the closed one. So there is a glucose molecule, and in the second closed one there is a glucose molecule which is inside these domains. So this is a closed figure which is being shown. So here the structure of the active site of an enzyme is flexible. So here the structure of the enzyme is flexible. So there are two types of groups that are present in the active site of an enzyme. One is the butt stressing group and the other is the catalytic group. The butt stressing group helps in supporting the substrate, whereas the catalytic group, they helps to explain the mechanism of the enzyme catalysis. So when butt stressing group comes in contact with the substrate, the change takes place in the active site, and these changes, they help to bring the catalytic group that is opposite to that of the substrate bonds that are needed to be broken. So the two model will help us to understand and describe the mechanism of the enzyme action, that how enzyme action is being taken place. So these are the two models which describe the lock and key and the induced fit hypothesis, which completely describe the how the mechanism of the enzyme actions takes place. Next is the factors that affect the mechanism of enzymes. So there are three factors that are responsible for affecting the mechanism of enzyme catalysis. So first is temperature, second is hydrogen ion concentration, and the third is the substrate concentration. So in temperature, what is happening? There is an enzyme catalysis which works in a very narrow range of temperature. So the optimum temperature is the temperature at which the enzyme shows the highest catalytic activity. So the optimum temperature can be defined when the enzyme is showing its highest catalytic activity. Anything above and below the optimum temperature declines the enzyme activity. Low temperature makes the enzymes inactive, whereas the high temperature can make the enzymes denature the structure of the enzyme. For example, if you want to draw a graph here, so here is a graph. So when the temperature increases, so this is the temperature and this is the enzyme part. So as the temperature rises, there will be a denaturation in the structure of the enzyme as well. Now hydrogen ion concentration. So as there is an optimum temperature required for the enzyme to function, there is also a optimum pH concentration which is required. So any fall or rise in pH, they reduces the activity of the enzyme. So some enzymes, they show good catalytic activity in acidic medium, while some show in the alkaline medium. So every enzyme, they has an optimum pH where their activity is the maximum. Now the enzyme catalysis also depends upon the substrate concentration. Substrate acts on the enzyme and then it is being changed into the product. So when there is an increase in the concentration of substrate, there will be increase in the velocity of the enzymes as well. So this is about the three factors that affect the mechanism of the enzyme catalysis. Next is the nature of enzyme action. That what is the nature of the enzyme action? So when there is a presence of active site on each enzyme molecule, the substrate they comes in contact with the enzyme, and then they form the substrate enzyme substrate complex, as it was being shown in the lock and key mechanism or hypothesis as well. So after a while, the enzyme substrate complex changes to enzyme product complex, and then product gets separated from it. So this way, the enzyme is not used in the reaction, but the catalysis cycle helps to explain the mechanism of enzyme action to the following points. So what are these points? These points are, first of all, the substrate gets bound to the active site. So that there is a substrate which gets bound to the active site. It was being shown in the diagrammatic representation of the lock and key hypothesis. And this induces an alteration in the shape of the enzyme. This was being discussed in the induced fit hypothesis. Now there is an enzyme product complex which is being formed, which is by making and breaking the bonds. So there is an enzyme which is being released away from the product, and it will be available again from a fresh batch of the substrate. So this is the nature of enzyme action. Now we will be discussing about the conclusion. So as we have discussed all this, we conclude that here, first of all, the definition of enzyme, that the enzymes, they are proteinaceous in nature, and they are involved in the catalyzing of various biological and the biochemical processes. We have discussed two models here, that is the lock and key hypothesis and the induced fit model. So we have discussed both of these models. So through this model we can conclude that enzymes, they have a basic active site on them, where the substrate gets bound to it and then changed into the product. So there will be a enzyme substrate complex which has been formed, and after this there will be a product which has been formed. So the end product here will be the product. So in this way, enzymes can be used again and again, and it can be used in the reaction also. So they can be used in the reaction also. Now we have discussed the three factors that affect the rate of enzyme action or enzyme catalysis. And what are these factors? These factors are temperature, pH, and the substrate concentration. So these are all the things about the enzyme action. These are some of the references. Thank you.